Man, I love dogs so much. I wish I were one. If only I had different DNA. Unfortunately, I have human DNA, which is very different from a dog's DNA. DNA is a microscopic substance that contains our genes, and not like the pants. Genes provide the instructions to make every single living thing on the planet. These instructions are different for each organism. Every cat, dog, bird, insect, plant, they all have DNA. But what is DNA? DNA is made up of 4 bases, which are arranged into a code that your body reads to build you. Think of a Lego instruction manual. When you read the manual, they tell you to put this piece there and that piece there until you end up with a fantastic Millennium Falcon. Well, it's the same for your DNA and your body. Your DNA says to put this cell here and that cell there to build up your lungs, hearts, bone, and skin until you end up with your human body. These instructions help create everyone. In fact, all people on Earth share 99.9% of the same DNA. That 0.1% difference gives each person their own hair color, eye color, and own unique traits. Let's explore how we can extract DNA and observe it for ourselves. For this lab, we will need the following set of materials. Before you start, put the rubbing alcohol in a freezer so that it is cold enough for us to use later. Now, put a couple of strawberries into the Ziploc bag alongside 100 milliliters of distilled water. Tap water is okay to use, but for the best results, distilled water is recommended. Next, throw in a couple shakes of table salt into the Ziploc bag and a dash of dish soap. Close the Ziploc bag tightly. Now, without breaking the bag, start mashing up the strawberries inside. When we combine the mashed up strawberries and salt with soap, the cell walls of the strawberry are broken up, while the DNA is kept intact with the help of the salt. This allows the DNA, which is contained inside the nucleus, to escape. But why does this work? Well, if we take a closer look at the structure of the soap molecules, we're able to see that it's made up of 2 parts. One side is attracted to water, meaning it's hydrophilic, while the other is attracted to fat, or hydrophobic. When these molecules come in contact with the strawberry mush, the hydrophilic side bonds to the water, while the hydrophobic side bonds to the walls of the nucleus. These 2 forces work together to pull the nucleus apart and release the DNA inside. The salt makes sure that the DNA stays together, and the rubbing alcohol tangles up the DNA into a gooey pulp, allowing us to observe it. Once it is thoroughly mashed up, leave the solution to sit for a couple of minutes. Then, slowly pour the solution over a strainer that is placed on top of the bowl. Now, get the rubbing alcohol from your freezer and pour in about 50 to 100 milliliters. Lastly, use a toothpick to fish out the white goop from the solution and place it on a piece of paper. This is your strawberry DNA. I hope you enjoyed today's lab, where we all took our first step into understanding what me, you, and all living things are made from. Now that you know how you can extract DNA from any living cell, happy sciencing!